started for today. All right, so we're gonna bring in everything together. That's the goal. So remember, everything we're building around is gonna be around the goal of dogma, right? So remember, central dogma, keywords I want you guys to know for this part, right, is we're looking at 3 different levels of molecules, right? We're going from the DNA, RNA, and then we're going all the way to the protein, right? You gotta feel comfortable with those 3 molecules. Within that, you guys remember that there's big steps, right? So the 2 big steps are we have our RNA synthesis, which is the first part of gene expression, from DNA to RNA. And the next step is we're gonna get from RNA to protein, which is gonna be our translation. Right? So transcription, translation, these 2 words. Feel comfortable with them. Right now, it's kind of set up as we go through the slides. We're basically going to be working our way through the different molecules, the stuff we see colored outside, of how we can regulate these different molecules. Right? There's different ways. In the next picture, I kind of have 2 different drawings, right? Arrows means that it's a positive signal, so something that activates. These little flat arrowheads are basically things that can stop it or inhibit it. So that's the big question. That's the big goal of the lab. I'm gonna put this off to the side as my key. Make it better afterwards. Now I can clean up one more slide. All right, one less slide. So now when we look at this and we're looking at, hey, how is this stuff being regulated, right? Remember that I like the way to start all the way at the top is what's going on with the DNA itself, right? So for this, you guys should feel comfortable with these histones and the fact that they can get modified. The modifications can be either positive or negative. My favorite acetylation, if you don't remember it that well, I'm okay with it, just as long as it gives me some idea that the histone and the modifications affect how the DNA coils, aka either it's open or it's closed. The next level we're going to jump into is if the DNA is open, then the big players there are going to be our enhancers and our transcription factors. Within this group, I have a positive and a negative arrow because there's 2 gangs of them, right? Like 2 different groups. Both of them bind to the DNA. That's something they share in common. The thing that they separate with is the activators, which turn genes on. They normally can help activate transcription, or they can change the histones. The negative regulators that either block the polymerase or block the RNA function in one of these later steps, or it can affect the histone and close the DNA. Right. So there we have these 2 roles that we can switch back and forth. Once we get a little bit closer, we're going to get into this other group of proximal promoters of the DNA, general transcription factors, GTFs, are basically all the machinery that help you clamp onto that last piece of DNA right before you get to the gene is coded, and they guide the polymerase. They help it sit at the right spot, and then it fires off. Once it's fired off, then the next big player is going to be the 4-2. Right? That's the molecule that we care about. It's the one that's going to start making our mRNA. And for that one, I want you guys to feel really comfortable with the C-terminal. So remember for that one, the big thing there is phosphorylation, phosphorylation, phosphorylation. In the beginning, phosphorylation turns it on, aka the general transcription factors are holding the DNA, the polymerase lands, and then in order to get it away from here, right, you already have this interaction holding it in place. The phosphorylation makes it like unstable, it doesn't want to bind well with these partners anymore, and that's what releases what helps it go forward. As it's doing that, the phosphorylations also help with 5' capping, they help with splicing, they help with the poly A, right? So the phosphorylation signal tells it to work. It can also give extra information to keep going. Next step to that is you've already made your mRNA, you put the 5' cap, you made it more stable. You put up the poly A tail, you made it even more stable depending on how long you make it. Then you can have something, a little bit regulation where the RNAi, remember this is a small piece of RNA that goes, finds our mRNA of interest, and you can turn it off 2 ways. Either it cuts the RNA, or when it binds, it basically blocks the ribosome from trying to make that into protein. But either way, it's a way to kill off gene expression. Then here's the more recent stuff that we've been talking about lately right now, right? So first step is, let's get the RNA, if it didn't get cut and it's still there, how do we make it? For this one, I need you guys to feel comfortable with tRNAs and your rRNAs, right? In this section, and like the one right above it, please remember to bring in your initials before RNA when you're writing about it, yeah? tRNA is the one that has your codon, anticodon. Remember, it helps you figure out what mRNA you have. At the same time, it also has the specific amino acid attached to it. So we can translate RNA code into a specific amino acid, and we can make them in the right order. For this one, I want you guys to be comfortable with these three guys. EIF, EEF, and then the release factor, right? The big key thing here is we're looking at eukaryotes, that's the first E. The one you should care about is this I or this E. So these are our initiation factors. When we look at the initiation factors there, the goal is we have an mRNA, and we start bringing everything together. So those EIFs are going to bring mRNA, our first tRNA, and it's going to bring the small cell unit of the ribosome. Right? It's going to help build that first complex. Then you bring in, once it glides down the mRNA, finds the right target, you can bring in the big one, and that's how you initiate. After that, you go through several rounds of these elongation factors. There's 2 of them. The big job there is the first one helps the tRNA make sure that it's matching up into the right sequence, codon and anticodon, and the pockets. Once it's found, it helps catalyze the reaction of changing amino acids to the growing chain. At the same time, afterwards, the next factor helps move the whole ribosome across the RNA to find the next target, and it shifts the pocket. We'll go into a little bit. And then the final one is after multiple rounds of elongation, you finally get to the release factor, which is it recognizes sequences that help end translation. At this point, it grabs onto the anticodon. The anticodon recognizes the codon, but it doesn't have an amino acid, so it can't catalyze the growing chain, and the whole thing literally falls apart. Right? At this point, you've made your protein. Here I'm gonna go a little bit into detail. Then we're gonna go to this part, right? You have your protein, it's made. One of the things is some of these proteins, in order to enhance their function, to direct their function, or to move them to a specific compartment, we give them modifications. Yeah. For these, the ones I want you guys to kind of look into or be comfortable with is the phosphorylation. Ubiquitination is another good one, right? This is linked to destruction. And then these sugar molecules. Glycosylation is important because we're going to bring it up a little bit later about how we fold the protein. I'm going to go a little bit to the chaperones and likely into proteolysis, which is just how we cut the proteins. Right? In that case, remember, you're just modifying it to get it to the final shape, get it working, it's going to look good. On the other side is you have this final checkpoint, which is either if the protein isn't properly folded, or if you want to get rid of it from the system and its life has ended. In that case, when it's time to clear off the protein, you're going to be thinking about this ubiquitin. Remember, that's the tag that gets added to the amino acids. Once it builds up, we send it to the proteasome. The proteasome literally grabs it and just starts trimming the amino acids. Another case is if we make the protein not folded properly, it's behaving. The big thing I want you guys to get from this section is we have hydrophobic residues that are sticking out. Right? In your head, I want you to think it's like if your lung was hanging outside your body, people around you would react, right? AKA your internal organs are hydrophobic. That's what I would imagine. But here, feel comfortable with these 2 programs. We have our ER associated degradation, and we have this ubiquitin unfolded protein response. For this, that's the goal. You're going to see that they both have mechanisms that once you have misfolded proteins, there's going to be some modification happening or some signaling response that we're going to activate in order to try to fix the situation. For this one, I expect all you guys to feel comfortable with ERAD. With ERAD, remember the big key molecule there is the glucose, right? So you know glucose, that's the one that you care about. When we get to the UPR, I'm still working this question myself, trying to work it around. I'm gonna bring this for sure. I'm gonna bring this one up. There's 3 different versions or 3 molecules that get activated. Right? You have the IR1, ATF6, PERK. Pick one and know it well. Right? In there, I want you to really take note of each one of this has really cool mechanisms from the stuff we've talked about before. Right? So you guys remember the IRE1 that one, the proteins, they phosphorylate each other to activate each other, right? Cool modifications there. Then they behave as spliceosomes, right? They'll cut RNA to make a new transcription factor, right? Showing cool mechanism up there. That new molecule they make now behaves as a transcription factor, goes into the nucleus, expresses the genes required to help build proteins. Then literally goes all the way to the top and gets this thing going down. That's why I really love these, and this is gonna be the big question at the end, because it's gonna let us connect a specific folding pathway, we're literally connecting into this thing up here. Just how this one works, you guys can go through the ATF6, it has the same thing, await detection. This one turns on kinases, the kinases through the cool mechanism of what we brought up, EIF2, it literally stops translation. That's how cool it is. Right? Stops translation. Again, also activates the transcription factors. This guy's the simplest one, which is the transcription factor is literally there, sitting, waiting, trapped. The moment the unfolded proteins show up, it literally will kick them off. That molecule then gets moved to another compartment, and it goes through what's called proteolysis. Right? It gets chopped up, and now that it's not attached to a membrane, this part acts as a transcription factor. Goes binds to DNA and literally turns on another set of proteins. So, big stories here. But pick one of these, you'll be good. And I want you guys to be super comfortable with this one. Focus on glucose. Yeah? All right, cool. That's the goal. So, regulation. For these, again, if you don't remember the molecule, exactly what it's called, you have a rough estimate of how you think it sounds, write it in. If you don't even remember exactly what it is, like write your best guess and give me like a description of what the function it does related to what you're trying to show off. Right? So as long as you give me that, I'll be able to give you some half credit at some point. That make sense? Cool. So I broke them down, the slides by each lecture. My goal here was to kind of like put out the key slides. Some of these I'm still going to clean them up a little bit. So for the first one, it was like, when we're looking at eukaryotic and prokaryotic cells, or how the cells work, just to compare them, right? The big thing is prokaryotes, the basic, right? They got a plasma membrane, everything's inside. Your DNA, your ribosomes, your mRNA, it's all one big compartment, everything's happening at once. On this side, this is where we're at, eukaryotes, we're fancy, right? We got our first plasma membrane running everything else. On the inside, we have multiple organelles. The ones I care about are the ones we're gonna bring up a lot, right? Nucleus. That's where we keep our RNA, and that's where we first make, keep our DNA, and that's where we first make the RNA. The next one, the one we bring up a lot in the class, is gonna be the ER. That's where translation is happening, right? We're making RNA to protein. The other one is, that's when we first start making those sugar modifications, right? Those key things about protein. And then the last one that we're gonna go into is gonna be the protein, right? Where it's like, they modify it a little bit more. For the repair mechanism, if you bring it up, that's where the protein gets cut and makes it inactivated. So these, as long as you know that they're separated by the plasma membrane to give us different compartments, and we can send proteins into them to do specific tasks, that's the fancy stuff. These red slides, if they were my notes, I would delete them. Like, as long as I know those three things, I'm good to go. And again, the whole goal is I want you guys to localize the central dogma into what compartment is happening in, right? DNA, it stays in the nucleus, RNA gets made here, you move out to the ER. In the ER, this is where translation is happening. You make the RNA turn to protein, and normally it goes from the ER and it'll travel to your Golgi vesicle, and then eventually it can get destroyed. That's your pathway. As long as you have that big picture, I'm okay. Number two, so for this one, I'm getting a little bit more into how proteins work, how enzymes work. For this, like I said, I don't know how you guys study. I want you guys to try them out. One way is I kind of did these little summaries about what's the main big take-home message of that chapter or that section of the book. I'm still going to go back, and I'm going to add a little bit something like this, where anything that's important, I'll try to make it extra bold. If I think it's a minor point, I'm going to try to make it in gray like this, right? So if you want to read that, try to understand it as a story to guide you to go back to studying your notes or studying the book. You can use the text. The other one is, one of my friends really liked to study with flashcards. So the way he studied is he would pick out the key terms like this. Right now, they're not organized in any particular order, right? So what he would do is he would grab these, put the keywords in front, the definitions in the back. So you would first practice going through the parts and memorize them, and then after you memorize them, you can organize them into groups and how they work, right? So if you guys look at this, you guys can see, like, some easy ones are kind of like, that if you're trying to figure out like polypeptides, right? Like, okay, you're linking up amino acids. Part of that is like sub-definitions to this, or of course, are these four things, right? Primary, secondary, tertiary, ordinary. So for that, you can tell me, hey, primary amino acids in a straight row, secondary amino acids link up with each other locally, right? Like alpha helix, beta sheet. So these will be a subcategory of that. Tertiary is how like the R groups link up, and the ordinary is how they group. So you can kind of group these words to tell yourself your story. If you can explain to me a process or how these things link up, you'll be okay, because like a big portion of the test is free response. Right? You're going to be describing systems, or I'll tell you how something's broken, and you got to guess what could be wrong. Yeah. So this is another way for you guys to look at it. So for this one, it's going to be like two parts: how proteins fold, emphasis on these hydrophobic, hydrophilic. You know, you guys don't love bringing that up. And then the other one was we talked about enzymes. So for this, they have their own vocabulary, right? Substrate, product, right? You'll have your energy of activation and your delta G. Put them into your story, like organize it into either a map or your flashcards, however you want to study it, chase that one down. So for that, I have my notes here, right? I started with the amino acids. The basic thing here, I might hit this just a little bit, right? Being like, oh, wow, what's the structure? But you guys know that what I really care about is this over here, right? I don't care that you guys memorize the amino acids. I'll give you the table. The thing that I want you guys to know is, are they hydrophobic, are they hydrophilic? Because that's going to affect how they interact, especially when we talk about things downstream. So hydrophobic, we'll stick with hydrophobic. And then these two, things that have opposite charge will pull into each other, right? They'll be hitting like magnets. Or you'll get hydrogen bonds. That's the big thing. I'll put my little key here. So I brought that there, they should be fine. So let's go. Again, a lot of things is the protein is where we get the job done, right? They're the hard workers, they're the one to carry up all the tasks. So whenever we're thinking of enzyme doing something, something's getting cut, something's getting glued, we're thinking of their behaving like enzymes, right? They're trying to catalyze some kind of reaction. So these are the basic rules that we're following. Feel comfortable with those, right? So they'll help make it easier to happen. They can't force it to happen. They don't make it using extra energy. Feel comfortable with the rules. And then, again, learn their key vocab here. Delta E, delta G, and then substrate and product. I might throw in one of these charts just to give you guys, analyze the data for me, yeah? And then this one, it's a really good example of why I think it's so important, right? I think some people had this one wrong on the quiz that we took, which was your enzyme, nobody have, like in my head, I almost think all of them have like two jobs. One is a binding pocket, which is where you grab onto what you're gonna work with, and then the other thing is your active site, which is where you do the function, right? So maybe like the cutting. In this case, I'm just showing you two different enzymes, chymotrypsin and trypsin, right? We're basically zoomed in to a specific part of that coenzyme, which is the colored area, right? This is this protein, this one, this protein. In this case, in this region, this one would have a bunch of hydrophobic amino acids. So it makes it so that if this was the chain of amino acids we were trying to digest or cut or catalyze, as long as you have enough in a basic amino acid, this will be the place that we can grab onto. Right, so binding pocket just grabs on. Again, on this side, positive, negative charge. So negative charge on our trypsin, positive charge on the protein we're trying to digest. But this is really just going to hold it. It's not involved in cutting, right? For cutting, you have to go into this thing. This was the active side, or where the function's happening. For this one, you can see this is still the same one from before. It showed in the green one, but it's a little purple on this one. Here is where we're holding the amino acids, so we know what we're grabbing onto. Then these two are the big players, right? 195 and histidine 57. These two amino acids are your active site. They're the place where the job's actually getting done. Right? You don't have to know these exact steps. I just want you to know that it's there. I put the slides red because if they were my notes, I would cut them out. That's all I care about, active site. I won't ask you specifically amino acids, but I just want you to know that they're apart from each other. There's two, each one does its own task. Everyone's cool with that? Yeah. All right. But if you go through it, It follows all the rules. It helps the reaction go forward. The enzyme that we start out with, the orange one, it's gonna look exactly the same by the time we're done. So this first picture here, will look exactly the same as the last picture down there, yeah. But binding site, active site, I want you guys to be comfortable with that. And the other one is, again, my favorite stuff, regulation, 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 right? So for this one, feel comfortable with these two different types of regulation. One of them is competitive and non-competitive. Competitive means that we're competing for that active site, things are being cut. Right, so in that case, it'd be like this little blue one's going to the binding site. If I was like, oh, I want to ask you about this one, you'd imagine a protein that blocks these two amino acids right here, would technically make this thing non-functional, right? But it would be through that competitive thing. If you're blocking the site, now you can't do it. The other way is this allosteric regulation, which means you bind somewhere else and you're going to change the way the protein is shaped. By doing that, you're going to either hide, block, or modify the active site. Sometimes you can even affect the binding site. Right? The whole goal is you turn it off by the enzyme itself won't work anymore. You know? So a lot of the stuff that we're going to be looking downstream, like transcription factors and all of them, they act like this. They're enzymes, but based on their partners, they might change whether they're able to work better, right? So, like the polymerase and phosphorylation, I could bring it up and ask you, like, if you're thinking about the polymerase as an enzyme, like how would you describe the phosphate function to it, right? So it'd be a, like, is it an activator? Is it an inhibitor? If it's an inhibitor, is it like allosteric? Like, what does it do? Yeah, all right. All right, number 3, we're gonna go into transcription. For this one, the slides, I set them up how they did it in the textbook, like we followed the textbook. So it's a little bit weird for mine because we just went from proteins and we're kind of working right here, right? The proximal promoters. Like how do we get the polymerase to start working? As a background, I put the prokaryotes, I put red on it to show you guys, like, I'm not gonna be too picky on this. I keep trying to put red on my slides. But again, they're basically a basic version of us, right? So when we think about them, they're really gonna have, the polymerase only has like 4 different proteins that help make the RNA, but the only thing that really guides them are these like sigma factors, right? So remember, for us, we have a bunch of general transcription factors. They literally have one, and they have very similar sequences. So for them, it's, they literally grab onto where it's gonna go, they put the polymerase, and they let it go, right? So they're a little basic compared to us on the other side, which is we have like a whole complex mechanism of having multiple levels of regulation, right? So for this one, for ours, here I give you a little bit of background of all the things our polymerase does. But the main thing here I want you guys to pull out from this slide is up here, basically the orange, like the little blobby stuff, that's our proteins. All of those are general transcription factors. We have things like this pattern-binding protein, this TDP-associated factor. They all basically stick to the DNA, they stick to each other with the final goal of bringing the polymerase. That's all they do, right? So I don't expect you to know all the names. I'm okay with you guys just saying general transcription factors. The other one is they bind to the DNA. They each have their own specific sequence, and that's what we call a proximal promoter, right? So that is in front of every single gene you want to make, you better have a proximal promoter so you can bring in all these transcription factors so the polymerase can do its job. Okay. You guys are comfortable with that? I'm cool with it. The next level, like I said, I need you guys to look into that polymerase. Once it's set up on that spot, it's going to be told how to fire off, right? And this is going to be coming, it's going to use information coming from the CTD, so the C-terminal domain, this little tail here. I have a little slide. Where didn't put it. Yeah. So that's the big thing, right? You phosphorylate the tail, it kicks it away from the whole complex that was holding it before. Right? So again, if I told you, thinking about like the general transcription factors, right? If our polymerase is an enzyme, how do the general transcription factors behave with it? Right? Like the active site, where it grabs the DNA, it's gluing DNA, they don't hold on to that, right? They're literally holding on to the back tail of your polymerase. Like they're holding it in place like this right in the body. The active site isn't, right? So they're kind of like allosteric inhibitors. They're holding onto it, they don't let it do its job. Phosphorylation comes in, that's like an allosteric activation, right? The CTD itself doesn't add base pairs. It doesn't hold on to your DNA. But once you add those modifications, it activates it, and now it can do its enzymatic function. Does that make sense? Can you prove that? All right. So I'm gonna try bringing questions to connect the stuff. So yeah, so remember my big themes, it's regulation, and the other one is like charge or binding of pockets and stuff. That's like my favorite things. All right, so everyone feeling cool with transcription for this center part? For this one, we get a little bit bigger, which is the enhancers. So it's like the stuff further apart. The cool thing about the enhancers is they're very specialized. So whatever gene you want to work on, you give them special enhancers. Depending on what can bind there, we can determine where and when we make the genes, right? So for this one, I put a few slides showing you the gene models, right? And the big thing here is like, there's a bunch of space outside the gene. That's where you hide your information, you know? So for this one, I have these two molecules, enhancers and transcription factors. The DNA they recognize, these are the molecules that turn it on and off, you know? I'm gonna, like I said, I'm gonna leave this one open-ish, and I'm hoping to mix it in with the question with the folded protein stuff that I talked about earlier, right? So remember I told you that unfolded protein response, or UPD, I always say it wrong, but there, all those molecules end up turning on a new gene. They're now the transcription factors, the genes that we care about, or all the molecules we're gonna use, they all have enhancers specialized for these factors, right? That's the big question. Like I'm gonna have you guys, I want you guys to basically explain it to me. Yeah. All right, cool. Yeah, here's the one. If we look into the polymerase itself, phosphorylation, phosphorylation, phosphorylation. Yeah. All right, cool. For the next part, it's gonna be the CTD. It also helps setting these things up. The things that happen to the RNA, the 5' captive splicing in the polymerase tail, as it's getting made, that phosphorylation tail is sending signals out to bringing in these enzymes that help modify our RNA. So when we're making it, as it's coming out, we're adding details to it, right? The cap, splicing, and poly A. For this, the big key things is that's protection. This, the longer the tail, the more stable, the more time you have. So it's an extension of life. The middle thing is the splicing. There, the big key one is splicing changes what we make. Right? So if we regulate that, we can make different proteins. Again, the connecting example is our protein folding stuff, right? They have a specific splicing that changes each job. All right. Splicing, I'm not going to it lightly, but you guys know that the big story is RNA helps target what we want to cut. It's not random splicing. We have a specific target. If you don't put small nuclear RNAs, as long as you explain to me that your spliceosome has RNA parts that guide it, I'll be okay. All right. Translation, how do you make your RNA go into protein? This one, again, I'm bringing up the basic ones, which is our prokaryotes, this thing on top. They literally go and they find these sequences that they're called Shine-Dalgamo sequences. And basically what happens is whenever they show up at the sequence, that's where they start translation. The cool thing about it is you can actually make one huge long RNA. As long as you put multiple sequences on it, you can make multiple proteins at once, right? So their stuff is, let's get the stuff produced as fast as possible and as much as you can. They normally group all their genes next to each other. We, on the other hand, are super fancy and we're like, we regulate one gene at a time. So for us, this is a ribosome, and we're gonna get into it right now. We have to go through an initiation step, which checks that our mRNA has a 5' cap, it checks that they have a poly A tail. And then once, and only then it'll start scanning it and going through it and actually making just one polypeptide, and it stops, all right? So for this, feel comfortable with the components, right? Your mRNA, your RNA, your tRNA, your amino acids, those are the big players. These, no, you get these, great. If you kind of get stuck on them and you see something, I just explain what they mean, I'll take it. For this one, you're gonna break it down to 3 steps: initiation, elongation, and your termination. In my notes here, I'll probably should add it here. The big thing that's gonna help us here is gonna be the EIF, the initiation factors, the EEF, the eukaryotic elongation factors, and this one I think is called like the terminator or something. So, let me see. for this, like I said, I drew in the really complicated thing, but the big story is all the EIFs right here in EIF 2, EIF 1, EIF 3. In this first part, what they're doing is they're bringing in all the subunits together to set this thing up. So if you just tell me EIFs are helping set up the first complex, it starts with your tRNA, your 40S subunit or the small subunit, and then finally bringing in the mRNA. Each one of these EIF factors, it has specific pockets to recognize its partner, but they also have affinity each other to bring the whole complex together, right? So that's the whole goal. Stick to their molecule of interest and then bring the complex together. Once you set it up, then you start looking for the first AUG. The critical one of this is where we start making the protein, right? Once you do that, their job of initiation transcription is done. You kick them all off, and you're going to bring in that 60S to replace it. And now you have this working functional ribosome, right? At that point, you switch over to elongation factors. And here, 2 jobs. I don't care if you forget the names, just know that there's 2 jobs. The first job is one of the factors helps find your tRNA. It also helps bring it into the pocket. If it matches the codons, it's the thing that helps you cut it off. So it lets you kick off the tRNA and transfer the protein over. That's this up here. The second part is once that's done, you got to shift this molecule over. EIF2 is going to give you that energy to move that molecule across that mRNA. So you're going to jump it 3 bases forward, right? And that's how you're going to just keep going back over and over the system. You have an empty pocket. EIF1 brings in the next tRNA. Once it gets glued in, right? EIF1 gets kicked out. And then you got to jump space 1 more time, opening up that first A pocket. Yeah, A pocket. That makes sense? Yeah? Remember, it's off on the names, just know the story behind it. And then finally, once you're done, you get all the way to the end, it's a release factor that helps us out. There's codons that we don't have a tRNA with a peptide to it. The release factor binds. Now that you can't have a growing chain, this thing isn't in the proper shape. It literally falls apart, right? So feel comfortable with these 3 steps and just know the 3 factors for you. Let me see. So I'm going through this really quick, guys. I know. RNAi, super easy, right? RNAi, the big goal here is it's RNA. It might start off, it's double-stranded. The goal there is you have 2 proteins. So know your RISC and your dicer. Those are the 2 big molecules. Dicer cuts it up. The RISC complex is the one that grabs one and literally goes and looking for any mRNA that matches with its partner. Right? The goal there is either we're going to cut RNA or we're going to bind to that mRNA. We're not going to let translation happen. As long as you guys got that, cool. Yeah. And then post-translational modifications. Again, here, the big one, I think you guys are going to like, is going to be phosphorylation. Again, it's going to come up with the UDP thing, right? We're going to bring it up again. By phosphorylating, you can change the task. The things that helps, either you have a kinase or a phosphorylation, which changes that phosphorylation status. But the whole goal is, using that mark, you can change whether it's working or not, right? Here, I'm just showing you a little bit of like, yeah, you can get a lot of modifications and stuff, depending on what domain you hit, you can change the task, but again, we're gonna care about is gonna be the UDP, yeah. The other one we talk about is gonna be sugar. I care about this on the right, what does sugar do to the proteins, like how are they helpful? I don't care about this. I was gonna try to put in a gray so you guys don't look at it, but you guys can know that all those changes happen there, but for the class, just know that they're getting modified, and this is the big things that sugars are doing, you know. But the main one, again, is going to be keep an eye on that glucose. When you're trying to fold the protein, glucose is the big key molecule. If it's properly folded, so the glucose helps it recognize the folding partner or the chaperone. If it's properly folded, it'll be sent out, it'll be fine. If it's not, it'll get another sugar molecule added back to it, and it goes through another round of trying to fold the protein. So that's where it's helpful for the sugar. And then the last one, I didn't put it here, but my other favorite one is ubiquitin, right? Which is you tag them with ubiquitin, you know we're going to throw them away. All right. This slide, I put this one in the beginning. I don't think you guys need this. So chaperones help with the protein folding. Again, this is the big story there is that when you're making the amino acid chain, you're going to have pockets that are hydrophobic. They're not going to be happy. And so these chaperones basically hold on to it so you're able to get the whole molecule out. then they can kind of leave and give it a chance to fold.
For the chaperones, is it every time a change is made, the chaperones are there, or is it only if like something wrong is detected that they're there?
Good catch, I was gonna skip it, but you have different types.
Oh,
Right. So
okay,
you have this one that's like, it's always trying to help, because some proteins are gonna need to be going through, or they have regions that are hydrophobic, that are clamped up to each other.
Mhm.
So those you always need to be helping them when they're getting beat.
okay,
You also need them when like, if you're trying to put a protein into the ER,
Mhm.
because it's a longer shape, you literally have to take it apart. You have chaperones holding it on this side, and you put it like a bead. So
okay,
you have to, again, hold on to the hydrophobic pockets to let it refold to your side. Then there's these guys, like the heat shock proteins.
Mhm.
This is, the protein's not folded properly. There's a hydrophobic region sticking out. Then they'll basically encapsulate it, and they'll use the amino acids of the chaperone to help fold the broken protein. So if you're properly folded, this is the one I carry, you can just leave.
Okay.
You wouldn't be part of it at all.
Okay.
So there's two, so good question. Midterm question, I like it. So now we get into the ubiquitin pathway here. For this one, remember, my biggest theme that I care about, big player, E3, that's my favorite, right? Yes, E1 helps charge up, getting that first ubiquitin to show up. It gives it to E2. E2 is held in place by E3. But E3 is doing a big job of bringing in the thing that's going to ubiquitinate and the target protein, right? So it's going to help set up that reaction so it happens. So to me, the big player is this guy, because it talks about recognizing specific proteins and how do you tag them with this ubiquitin tag. So that E3 is recognizing that something's wrong with this protein, and that's why it's tagging it. If this protein gets only a few marks, it might still be functional. If the protein doesn't have enough time to fold itself properly, the system keeps adding more and more ubiquitin to it. So eventually you get enough tags of ubiquitin, then we go to this big player, which is the proteasome, which is really cool. It's basically just like a tunnel. You put the proteins, the ubiquitin brings it to that spot, like it gets recognized from the ubiquitin tag, it gets thrown into that tunnel. Inside, the enzyme, basically the protein just starts cutting the amino acid chain, right, and shreds your proteins. So ubiquitin, proteasome, in your head, always think of those two together. E3 and proteasome are the big players, right? All right. This part. I want to make sure that these three slots, this one should be. So anything purple, remember that's my key for it. It's going to be on the midterm, right? This one, I care about it a lot. It has a really cool regulation to it, and it shows a little bit of, it has a lot of the key things about how the molecules are kind of like sensing each other, right? So here the big thing is, you should be trying to fold your protein, the thing you do, right? When it's coming in, it first gets tagged with sugars. The one that I care the most about is we're keeping an eye on the glucose, right? That's the big signal. This thing will try to recognize hydrophobic pockets, right? And it uses the sugar. And what it's trying to do is it's trying to help it give it a chance to fold it. After it does it, it takes off the sugar, releases it. If the protein's good, it can keep going, it's fine, right? If the protein's misfolded, you're gonna have a hydrophobic thing sticking out. That hydrophobic thing sticking out is gonna get recognized by this guy, your UGGT. So this is like the checkpoint. If he grabs onto it and says, he hold on to hydrophobic proteins, they're having their things hanging out. I mean, my head, I think like their guts hanging out, right? But it's like, they have a hydrophobic domain sticking out, the protein is not happy. What they do is they basically add a sugar. So this GT stands for glucose transferase. So it's adding a sugar back to it. Once you add the glucose back to it, it sends it back to work with the cow protein. Yeah. So in this case, we're giving that protein a chance to try to fix itself, right? If it can't, then we go down. So the pathway, which is basically, we're gonna get it and then kick it out of the ER, and it's basically we're gonna end up getting tagged with ubiquitin, it's gonna get destroyed. So it's like, give you a chance to fix it, if it doesn't work out, you're gonna get trashed. Yeah.
Is this the same as the glycosylation in the ER, or is this?
Yeah. So this is the ER itself. Right here, you're in the lumen.
Okay.
So right here where it's like, it's your ER, so you're inside. So this one works very similar to the one we saw earlier. This guy. So it's doing the same thing where it's like, hey, you have a glucose, you have your protein, this one tries to fold it, If it's behaving well, cool, you can leave, that's a functional protein. If it's not folding properly, something's sticking out, here's your glucose transferase, you can add glucose back to it, you can give it one more shot of going to this fold. But again, my favorite molecule, right, wink wink, is that glucose. Because that glucose, like if the protein's hydrophobic on its own, this thing doesn't know what to do, like it doesn't fit all the right pockets for it to work. Only when it has the hydrophobic stuff sticking out, and it has a glucose molecule, then it knows like, okay, now we're gonna give it a shot and try to fold it. So, to me, the glucose in my head, when I'm almost thinking, I'm like, it's almost acting like like if I was trying to do the experiment by myself, I'd be like, I think it's like an allosteric activator, right? So if you come in with a glucose, and I kind of meditate to help you fold, wants to work. If you don't have a glucose, I'm not even gonna work with you, Make sense? All right, cool. I can also make the same argument for this guy. And talk about your glucose transferase, right? If your hydrophobic regions are hidden in the protein, we're not even gonna interact. Right? But only if the hydrophobic stuff is sticking out, then this one will try to recognize them, and now it can do its catalytic job of adding the glucose. Yeah. All right. Cool. And then this is the other big one. Again, pick one pathway, whichever one you like, and know it well. Yeah. I want you to know it. What would happen? Explain to me what happened in both cases. In one case, I want you to think about what would happen if all the proteins are folded properly. What state would I find the different molecules, right? So, for example, the first one, RA1, you have those two receptors, the ones that's sensing on the inside for the misfolded proteins. If everything's okay, they don't interact with each other, aka they don't phosphorate each other. Right? So in my notes, I'd be saying like they don't activate the kinase function, they're not phosphorated. Because they're not phosphorated, they don't do this job. Right? The ribonuclease, remember they're like lysosomes kind of. Because they're not activated, they won't do that job either. So then technically, the mRNA that they normally modify should be intact. So this would be the same RNA. They would be apart from each other, not phosphorylated, and this thing would look like this. Because if it still has an intron, that protein doesn't work. Okay. That's my story down the misfolded. Then you follow it down the folded side. If it is misfolded, the misfolded protein's gonna bring this thing together. They're gonna kinase phosphorylate each other. Right? That's gonna turn each other like they're gonna make themselves work better. Or like if their catalytic function is to cut RNA, this thing will be like the allosteric activation, right? You activate it, now you splice the mRNA, now you can include the transcription factor, and now you can turn these genes on. Yeah. Everyone's cool with that? So pick your favorite and present it both ways. What would happen to these molecules if it wasn't there, and what would be if the misfolded proteins were there? Yeah. And then I think for this one, for you, I challenge you to do the middle one. I think you were talking about EIF was your favorite, right? The EIF factors or no?
I don't remember
You remember that one? All right, because I thought it was you who brought it up to me after class, and I was like, all right, you should do that. So the cool one with this one, which I think is really better, the best thing about it is if the binding happens, it phosphorates itself, but it actually just phosphorates the factor that already exists. And it's that EIF, right? Big key on the I. So it's the initiation factor. So if we go back here, right? EIF is the one that EIF2 is the one that grabs onto our first tRNA to start translation. So it's like if this if something is going really, really bad, you want to stop translation as much as possible so you can have time or have proteins to fix things around it. So it will literally stop everything else from working. Because that phosphorylation mark is the big signal to turn it off. I remember that was your question you asked.
Yeah.
So that one literally just has a big response where I'm like, no, we're gonna turn off all translation until we figure this problem out. So I challenge you to do it. But, yeah, so big stuff. That's what I want you guys to look at. I tried making my notes as best I could. The other one is somebody asked me for videos and stuff. They said they study a little bit better that way, looking at stuff in 3D. I tried making like one or two videos tops for each section that I could find. This took a lot of time to look for them. If you guys find better stuff, please share them with me and I'll update my slides. When some of them, they have regions that they talk about stuff in more detail than we did. So I do these little tiny bits where I like focus on these regions, right? And then the other one is when they go over keywords that I care about, I kind of been typing them up here on the right side. If you study off the videos, the story isn't as complete, but in your head, if you bring up, like the way I was thinking was, you bring up your vocabulary list for that chapter, like this stuff, and it's like, could I fit all these words into this story or into the video? That'd be one way I can sit around. Yeah. So I don't know, have fun studying. I don't know if the slides are gonna help. I don't know if the little summaries are gonna help, the word list will help. Be creative and then let me know. That way next time I'll do a lot less. Like, any questions? No? All right, anything you guys want me to focus on when we meet on Friday?
Can you focus on the, were you know one of the three for the, what was it?
Oh, this guy.
Yes, yeah.
Yeah. Bring this one up again? Okay, I'll bring this one up. Anything else?
UPR.
Yeah. All right. Do you have any favorites? Questions? Sounds good. I'll take care of it. I'll do those two. Then, papers, I've only approved 2 or 3 papers out of everyone in the class. So if in your group, I sent you guys emails, right? Yeah, emails? You didn't let me know, but you should meet up with your group and then give me a paper right, hopefully by the end of today or tomorrow. Then you start reading them, so, yeah.
What did we get?
Yeah, they think your group had like 5 or 6 good ones. I put stars on it to be like, they're good. You guys can-
But if you emailed us and said this paper's good, we're good. I think you emailed our group.
I think so.